And what this is going to be a description of how Holliday junctions are resolved. So I'm starting out with a diagram showing the formation of two Holliday junctions following a double-strand break in the blue chromosome, and the formation of the Holliday junctions by forming a D-loop in conjunction with the intact red chromosome, which did not have the double-strand break. I've colored the DNA carefully according to the sequences that were copied, so that when a sequence was copied from the 3' overhang of the blue chromosome, for example, right here, but it was copying off of the red chromosome, I've drawn in red. And similarly down here, where there's a 3' end for the blue chromosome, that was then used as the primer for synthesizing DNA from the red chromosome, so that's all in red. So this is two Holliday junctions. Now, Holliday junctions can be resolved by making cleavages, because these two chromosomes are now attached by the DNA at two different locations. And so one way of making the cleavages is by making two longitudinal cuts. So I'm going to draw in a cut here. Okay, that's a pair of longitudinal cuts, and I could draw a pair of longitudinal cuts here and here. And if I do make those cuts, then all I have to do is take the 3' end of the blue chromosome up here and join it to the 5' end of this red piece of DNA. So that as a result of those two cuts, I am now going to have the following. So we're going to draw that in. And I'll have the red chromosome, and then the red DNA, and now the blue DNA. This is the 3' end, 5' end. And now I'll put in what happens to the upper strand, upper DNA here, the upper piece of DNA. I'm going to have the 3' end over here, and we're going to come over here, and we're going to come up to a cut on that strand, and that cut will allow ligation at this point. Let's continue with the cuts at the other Holliday junction. So this bottom strand on the upper chromosome is going to end up being joined right at this point. Okay. After those cuts. Now let's draw the resolution of the Holliday junction and how it affects the bottom chromosome. And as you can see, I'll draw this for you. The bottom strand of the bottom chromosome, of course, is intact. Nothing was ever done to it. But the upper strand, of course, after the cuts, what will happen is red DNA will get ligated to red DNA. There may be a little bit of blue here, but then that'll be ligated to the red DNA. Just so. Okay? So the bottom strand of DNA here is going to be virtually completely red. So the original intact DNA has been restored after the resolution of the Holliday junction. The difference is the upper strand, the upper DNA molecule, now has been repaired by copying from the intact chromosome, and that's the areas that were copied are shown in red here. And this kind of resolution of the Holliday junction actually converts some of the sequence in the blue chromosome to red sequence. And if the sequences aren't exactly the same, you're going to have a slight difference. Of course, since this is homologous recombination, the sequences are pretty close to the same. But there will be some differences depending on whether the blue chromosome and the red chromosome have exactly the same sequences. And because they're slightly different, we call this process here gene conversion. So the damaged chromosome has undergone a gene conversion. All right. Now, there is a different way you can resolve the Holliday junctions, and we're now going to talk about that. So I'm going to erase this way of repairing, resolving the two Holliday junctions, and show you a different way to resolve them. So let's get the eraser. Get rid of gene conversion here. And let's see, I'm going to be judicious in how I do this, because I'm going to make one pair of cuts the same. And so one pair of cuts will still be these longitudinal cuts that I drew previously. But the other pair of cuts are going to be transverse cuts. And here are the transverse cuts. All right. Now, as a result of those transverse cuts, I'm going to have a different species. So let me erase things here. Let's see. Let's clean this thing up a little bit. Let's get rid of that. Now we're going to make these transverse cuts, and I'm going to show them to you here where the cuts are. So imagine that this enzyme here nicks the DNA. Boom, there it goes. And boom, there it goes. Let me restore that arrow so you can remember where the cut was. There you go. And so that is where the transverse cuts are. Now, keeping track of the 3' and 5' ends, all right, there's going to be a 3' end over here, okay, and a 5' end on this side of the cut, all right, and there is no cut here, so this is still okay, and there is no cut over here, so this is still okay, all right. But there was a cut down here, so I'm going to redraw it for you, drawing in the 5' and 3' ends. So I've made these two cuts. I have 4 single-stranded ends, and those have to be rejoined in a recombinational way. So this 3' end is going to end up joined to this 5' end. And this 3' end is going to be joined to this 5' end. So let me denote that in the following way, just so we can keep track of bookkeeping. I'm going to make a triangle here because this 3' end is going to be joined to this 5' end. That's a triangle, and it's 2 red pieces of DNA. Okay, and then I'm going to make a circle over here to denote which 2 ends are going to be joined. So the circles will be joined. One circle from the red DNA and one circle from the blue DNA. So that's how these things are going to be joined. So now all we have to do is redraw this sort of unfolding things. And the way we can unfold things is we can flip this strand and this strand over. And let me just draw an arrow for you so that there's going to be a flip between these two. And I'm going to show you how that looks as a result. What you're going to see now, this is the first half, and then this is going to be that triangle on the upper strand. That's going to be joined to the triangle on the lower strand after the flipping. Okay. And this piece of DNA has been flipped over at this end. This is intact. Nothing has happened to it. So that's the 5' end. That's now the new upper DNA. And now the lower DNA, I'm going to draw in here. And we'll end. And here's the 5' end of the lower DNA. All right, remember that's going to be joined to the circle on the upper strand, so I'm flip that over to the blue DNA. So I'm going to actually make a little blue circle so you can see they came together. All right. And actually, let me draw it just so you can see that there are 2 triangles here and 2 circles on the bottom. So now you have 2 chromosomes that have been restored, and they've been separated by resolution of the Holliday Junction. The resolution occurred because of a longitudinal cut at this Holliday Junction and a transverse cut at this Holliday Junction. And as a result of the combination of the longitudinal cut and the transverse cut, okay, what I end up with is a chromosome, an upper chromosome here that starts out blue and ends up red, and a lower chromosome here that starts out red and ends up blue. And so that is not an easy thing to draw, so we have to congratulate ourselves and say voila. All right. What is this product called? This product, this method of, or this particular way of resolving the 2 Holliday junctions, these products are now called a crossover. So I'll just write out crossover from homologous recombination. Okay, that was very complicated, but if you keep track of the 5' and 3' ends of the DNA, you should be fine. So thank you very much.